So back in 2007, scientists basically figured out how to build a time machine.
Yeah, but not the sci-fi kind.
Right, no, not a metallic box that transports human beings through the centuries. I mean, a microscopic time machine for human cells.
Exactly.
They discovered this really highly complex biochemical method to take an aging, fully specialized adult cell, like a skin cell, for example, and just force it backward in time.
Which is incredible. They stripped away its identity.
Yeah, they erased its biological memory and essentially reverted it to a blank slate, like a biological master key that's capable of building virtually any tissue in the human body.
And today, we're looking at a text that takes that reality and asks a deeply provocative question.
Oh, absolutely. It asks, what if this exact type of cellular extraction and regeneration procedure was actually described with, surprising biological precision over 4,000 years ago?
It is a premise that really challenges how we categorize our knowledge.
Right.
We have this tendency to firmly separate the realm of faith and ancient texts from the realm of empirical metrics and laboratory science.
Like they're two completely different planets.
Right. We just assume they speak entirely different, incompatible languages.
Yeah.
But the material we're analyzing today argues that beneath the theological poetry, there lies a very literal, very tangible genetic reality.
Now, if you are listening to this right now, you are the kind of curious mind looking for a shortcut to being well-informed, right? Without drowning in the sheer volume of information out there. And today, our mission is to explore this really fascinating synthesis.
It really is unique.
Yeah, we are bridging cutting-edge cellular biology with ancient biblical theology. And our sole source for this deep dive is Chapter 8 from Ozair da Silva Mota's work, Analyses Biblico-Scientificos.
The specific chapter is titled Stem Cells, the Code of Life in the Formation of Eve.
Okay, let's unpack this. Because right out of the gate, we need to establish for you guys that this isn't just a philosophical thought experiment.
Definitely not.
The author is heavily suggesting that modern genetics, like the absolute bleeding edge of our understanding of human biology, might actually offer a literal blueprint for the creation of Eve from Adam's rib. But to even entertain that, we can't just skip straight to the theology.
No, you really can't. If we connect this to the bigger picture, you cannot grasp the theological argument the author is making without fundamentally understanding the microscopic mechanics of the human body first.
Right. The biology is the canvas.
Exactly. The biology is a canvas. And to understand that canvas, the text grounds its analysis by starting chronologically with, honestly, one of the most monumental scientific milestones in human history.
Which was June 26, 2000.
Yes. That was the day the world's leading scientific powers announced the completion of the Human Genome Project.
And I think it is crucial to understand what completion actually meant in this context, right? Because before the year 2000, we obviously knew DNA existed.
Sure, we knew the double helix.
Yeah, we knew it possessed the instructions for life, but it was essentially a locked vault.
That's a good way to put it.
The Human Genome Project wasn't just about acknowledging that the vault was there. It was the painstaking process of sequencing roughly 3 billion base pairs of DNA.
3 billion.
Yeah, 3 billion. It was humanity mapping the exact molecular syntax that actually builds a human being.
And the scale of that achievement is precisely why the text compares it to landing on the moon or, discovering new continents.
It's that massive.
It is.
Yeah.
It was an expedition into the microscopic inner space of our own existence. For the first time ever, biology was definitively understood as an information technology.
Wow, an information technology.
Right, because we weren't just looking at tissue anymore. We were looking at code. In fact, the source includes a specific quote from then-President Bill Clinton from that very announcement.
Oh, I remember that.
Yeah, he referred to the mapping of the genome as the unveiling of the script of life written by God.
It is just a profound way to frame it. You sequence the 3 billion base pairs, and suddenly you have the alphabet of human existence. It's like having a multi-trillion-page instruction manual, and we finally learned how to read the alphabet.
Exactly. But knowing the alphabet is just the prerequisite.
Right.
The text quickly pivots from the broad mapping of this genetic script down to the specific characters constructed by that alphabet. And in this biological narrative, those characters are the cells.
Right, and the author makes a very stark distinction here regarding the complexity of those cells.
He does. When we look at a simple unicellular organism like, say, an amoeba or a bacterium, we are looking at a solitary entity that must perform every necessary function of life on its own.
It's a one-man band.
Literally.
Yeah.
It digests, it respires, it reproduces, all within a single cellular membrane. But a human being. The text conceptualizes the human body not merely as an organism, but as a vast biological metropolis.
I love that phrase, biological metropolis. Because in this metropolis, you don't have solitary generalists. The text notes that humans are constructed from roughly 216 distinct types of highly specialized cells.
216 different professions, basically.
Yeah, required to keep the city functioning. Now, if you're listening, you probably know the basics. We know that red blood cells are the couriers transporting oxygen through the vascular highways.
Right.
We know neurons are the telecommunications grid, firing action potentials to process stimuli.
And muscle fibers are the biological motors handling physical contraction.
Exactly. But what is truly fascinating here isn't just what these cells do, it's how they become so specialized in the first place.
This is the key.
Because every single one of those 216 cell types contains the exact same sequenced genome, the exact same 3 billion base pairs.
Every single one.
So if the DNA is identical across the board, specialization isn't about what genes a cell actually possesses, right?
Right.
It has to be about what genes are actively turned on or turned off.
That is the crux of the biological mechanism. It is governed by a process called epigenetics.
Epigenetics.
Yeah. While the author uses fairly accessible language to describe this specialization, the underlying science is that a neuron and a red blood cell have the same DNA, but they have entirely different epigenetic markers.
Which are what, exactly?
They are chemical tags, like methyl groups, that physically bind to the DNA. They act as molecular, do-not-read signs.
Oh, wow.
So as a cell matures and specializes, it undergoes this massive locking down process. The genome is essentially folded up and silenced, leaving only the specific genes required for that cell's particular profession accessible.
It is an incredibly efficient system. But the text points out that this efficiency comes with a pretty severe biological trade-off.
It does.
It highlights what we could call the burden of specialization. Because these cells have folded away and silenced the vast majority of their genetic code, they become what biologists call terminally differentiated.
Meaning they are the final product.
Right. The epigenetic silencing is so profound that a specialized cell entirely loses the ability to transform into anything else.
Yes. A red blood cell will never regain the ability to become a muscle fiber.
It's locked in.
Completely. A neuron cannot suddenly reboot itself and decide to function as an epithelial cell in the skin. They are permanently locked into their biological destinies by their own epigenetic architecture.
So what does this all mean for the survival of the organism? If every worker in the metropolis is completely, permanently locked into their role, what happens when the city takes damage?
That's the big question.
Right. If a person suffers a severe hepatic injury, like their liver tissue is damaged, how does the organ actually repair itself? If the remaining liver cells are sometimes too damaged to replicate, and no other cell type in the body can switch jobs to help out.
That is the precise biological vulnerability the text raises, and it leads directly into the elegant solution established by the creator, according to the author's analysis.
Okay, lay it on me.
Well, for a highly specialized biological metropolis to survive, there had to be an innate backup system, a biological reserve that bypasses this terminal differentiation. And that reserve is the stem cell.
The stem cell. The source material uses a very evocative term for them. It calls them mother cells.
Mother cells. I like that translation.
Yeah, they are described as the raw biological material of the body, the fundamental matrix of reconstitution.
And their defining characteristic is their capacity to divide and differentiate. They are the blank slates.
Because they haven't been locked down yet.
Exactly. They have not undergone that massive epigenetic lockdown. Their genome is still wide open and accessible.
So let's drill down into how this actually works. How does a blank slate know what to become?
It's a highly sophisticated process.
Like, if we stick with the injured liver scenario, how do the mother cells even know they are needed, and how do they know they need to become hepatic tissue and not, say, bone cartilage?
The mechanism relies on highly complex biochemical signaling. When tissue is injured, it doesn't just passively fail. It actively sounds an alarm.
Like a biological SOS.
Literally. The damaged cells and the surrounding immune cells release specific proteins, chemokines, and inflammatory markers into the microenvironment. This creates a chemical gradient,
Okay.
and the stem cells, which reside in specific niches throughout the body, have receptors on their surface that actually detect these chemical distress signals.
So they essentially follow the chemical breadcrumbs straight to the site of the injury.
Exactly. It is a process called chemotaxis. They migrate right to the damaged area. Once they arrive, the microenvironment of the injured liver bombards the stem cell with very specific growth factors.
Just telling it what to do.
Right. These external signals bind to the stem cell's receptors, which triggers a whole cascade of internal signals that travel all the way into the nucleus. And these signals tell the stem cell exactly which genes to keep open and which ones to begin silencing.
That is wild.
Through this precise biochemical dialogue, the mother cell divides and differentiates into the exact specialized cell needed to reconstruct the tissue.
It is just a microscopic marvel, a literal promise of renewal embedded directly into our tissues. And this, right here, is the moment in the text where the biological reality acts as a springboard for the theological premise.
This is the pivot.
Yeah, the author posits a really direct question. If we know, as a matter of empirical scientific fact, that these mother cells possess the inherent capacity to reconstruct complex tissues and whole organs, could they be the biological vehicle utilized by the Creator?
It's a fascinating leap.
Right. When the ancient book of Genesis describes the formation of a completely new, whole human being from a physical fragment of Adam, are we looking at the theological description of a massive, perfectly executed stem cell procedure?
It is a staggering proposition.
Yeah.
It requires us to look at a narrative that has been interpreted as, pure divine magic or metaphor for millennia, and instead examine it through the lens of genetic mechanisms.
Which is not how most of us were taught this story in Sunday school.
Definitely not. However, before the text applies this theory directly to the biblical account, it insists on a deeper level of biological clarity. Because we really cannot understand the scope of this theory without understanding the distinct categories of stem cells. The text outlines what it calls a scientific triad.
Okay, let's walk through that triad for the listener. The first category the text establishes is embryonic stem cells, which are also known as pluripotent cells. And the source locates these at the absolute dawn of biological development.
Right. The text outlines the timeline of human development to pinpoint exactly where these cells exist. Begins with fertilization, obviously, the moment the sperm and the egg fuse to form a single-celled zygote.
Okay.
This single cell begins to divide rapidly. A few days into this division process, it forms a microscopic, hollow sphere of cells known as a blastocyst.
A blastocyst, right.
And inside this sphere is a cluster of cells called the inner cell mass. These specific cells are the embryonic stem cells.
And the key word the text uses for them is pluripotent.
Yes. Pluripotent means they possess the ultimate biological potential. They have the unrestricted capacity to differentiate into literally any of the more than 200 distinct tissue types in the human body.
They hold the master keys.
They hold the master keys to the entire biological metropolis.
But the text notes that this pluripotency is incredibly fleeting, right? It is a very tight window.
A very tight window. Merely 4 to 5 days, according to the source.
Wow, that fast.
After this brief period, as the embryo continues to develop and implant, these cells begin to receive their initial specialization instructions. They start that epigenetic locking process we discussed earlier, and they lose that absolute, unrestricted pluripotency.
Okay, so that is tier 1, the embryonic cells. Infinite potential, but an incredibly short lifespan in their pure blank slate form.
Correct.
The second category in the text triad is adult stem cells. These are classified as multipotent or unipotent. So where exactly do these reside in the human body?
The source explains that adult stem cells are maintained in specific tissues to handle the daily wear and tear of the organism. The text mentions the umbilical cord as a really rich source, but notes that they are predominantly located in the bone marrow.
Bone marrow, okay.
Yeah. These are specifically referred to as hematopoietic stem cells, which are constantly churning out new blood cells, and mesenchymal stem cells, which can generate bone, cartilage, and fat.
Got it.
Now, the critical distinction the author makes here is that these adult stem cells do not possess the ultimate master keys.
They are restricted.
Exactly. They are multipotent, meaning they can differentiate into a limited range of cell types related to their tissue of origin, but they cannot form every tissue in the body. They are restricted.
But they're still doing a lot of heavy lifting.
Oh, absolutely. They are entirely vital for our daily survival, and they are actually the biological basis for our currently established cellular treatments, like bone marrow transplants for leukemia.
Right, which we've been doing for decades. So we have the embryonic master keys that vanish in days, and the adult restricted keys that stay with us for life.
That's the baseline.
And then we arrive at the third category in the text, and here's where it gets really interesting. The author refers to this category as a lab miracle. These are Induced pluripotent stem cells, or iPS cells?
This is the time machine you referenced at the very beginning of our deep dive.
Yes.
The text highlights this as a profound scientific breakthrough that occurred in 2007.
And what scientists actually achieved in 2007 completely upends that burden of specialization we talked about.
It shatters it, really.
The text uses skin cells as an example. Scientists figured out how to take an adult, specialized cell and forcefully reprogram it. They successfully sent it back to an embryonic pluripotent state.
Which was previously thought impossible.
Right. Wait, so if the epigenetic locks are supposed to be permanent, if the DNA is heavily methylated and folded away in the filing cabinet, how exactly do you force a cell to forget its identity? Is it like a chemical wash? How do they unlock the genome?
It requires a highly targeted genetic intervention. The 2007 breakthrough, which was pioneered by Dr. Shinya Yamanaka and actually won him the Nobel Prize, involved identifying 4 specific master genes.
4 master genes.
Yes. These are genes that are highly active in the early embryo, but become permanently silenced in adult cells. They're transcription factors, meaning their entire job is just to turn other genes on or off.
Okay, so how did they get them in there?
The researchers use viral vectors, essentially modified viruses, to deliver active copies of these 4 master genes directly into the DNA of the adult skin cell.
Oh, wow. Using a virus to sneak the code in.
Exactly. Once inside, these transcription factors began a massive epigenetic remodeling process. They forcefully removed the methyl tags, they unfolded the chromatin, they systematically erased the cell's specialized memory, and reactivated the dormant embryonic code.
They biologically rewound the clock.
They completely rewound it.
It is astonishing. You are taking a cell that was absolutely definitively locked into being a skin cell, and by inserting these master switches, you revert it into a cell that can become cardiac tissue, or an optic nerve, or bone.
Anything.
And the text makes a crucial point about why this iPS technology is viewed with such massive hope in the medical community. It states that iPS cells represent our greatest avenue for curing degenerative diseases, precisely because they offer that unrestricted, pluripotent potential without triggering the massive ethical hurdles associated with the first category, the embryonic cells.
Right. And this brings us to a really pivotal section of the author's analysis. The deep dive navigates a profoundly complex ethical dilemma, which is often referred to as the ethical crossroads.
The ethical crossroads.
The text acknowledges that the ultimate goal of cellular therapy is universally noble, right? With healthy, vibrant tissue. It is the pursuit of healing.
Right. Nobody is against healing.
Exactly. However, the conflict detailed in the source arises because, despite the absolute miracles of iPS technology, embryonic stem cells which are harvested from actual embryos still currently yield superior, more stable research results in certain applications compared to the modified adult cells.
And as we navigate this specific part of the text, I want to explicitly state for you, the listener, that our role today is strictly to impartially report the contours of this debate as outlined by the author. We are not here to endorse a specific side.
No, absolutely not. We're just looking at how the source presents this inescapable moral collision.
Right. And the author frames the debate around one central, incredibly weighty question. When researchers utilize an in vitro embryo to harvest these highly effective pluripotent cells, a process which, destroys the blastocyst, are they merely disposing of unthinking biological material, or are they actively interrupting a human life?
That is the core of it. Is it cellular material, or is it a person? The text outlines 2 distinct viewpoints that clash at this exact intersection. First, it presents what it categorizes as the secular, or parts of the scientific viewpoint.
Right. Walk us through that framework.
According to the text, a portion of the scientific community views human development as a biological continuum, rather than a discrete, sudden event. They debate the precise onset of human individuality. The source notes that within this framework, the 3rd week after fertilization is frequently cited as a critical threshold.
The 3rd week. Why that specific time?
Biologically speaking, this aligns with a developmental milestone known as gastrulation and the appearance of the primitive streak. The argument is that before this point, the blastocyst can actually split to form identical twins. Therefore, some argue true singular human individuality cannot be established before this marker.
So under that specific scientific framework, the blastocyst at day 4 or 5, which is the exact window when embryonic stem cells are harvested, would exist prior to the onset of individuality.
Correct.
It would be viewed as potent biological material, sure, but not yet a distinct individual. But the text immediately juxtaposes this with the religious viewpoint, which offers a completely different definition of when life begins.
Yes, a totally different paradigm. The author outlines the Christian perspective, noting it is a view shared by various other religious traditions as well. This viewpoint unequivocally sustains that human life begins at the very moment of conception.
Okay, and what is the biological logic embedded within that theological view?
It centers on syngamy, the exact moment the haploid sperm and haploid egg fuse to create a diploid zygote. At that exact microsecond, a completely unique, never-before-seen human genome is established.
The script is written.
The genetic blueprint is finalized. Therefore, in this view, the blastocyst is not merely potential life. It is an active, living human being in the earliest stages of its natural developmental arc.
And the author masterfully synthesizes the weight of this tension. The text states the fear of, quote, killing lives to save others acts as the heavy moral brake on this science.
It is the core regulatory force.
Yeah, in societies that prize the sanctity of existence, you have a highest imaginable hopes for curing Alzheimer's, Parkinson's, and spinal cord injuries, colliding head-on with the deepest philosophical and theological definitions of what constitutes a human life.
This raises an important question, and it is the exact fulcrum the author uses to pivot our deep dive back into the biblical narrative.
Okay, here we go.
We have just spent significant time establishing the staggering reality of stem cells. We understand their power touches the very threshold of creation in a laboratory. We understand they are the fundamental microscopic code of physical life.
Right.
So if this is the undisputed biochemical reality of how human tissue is generated and regenerated, how does this precise scientific mechanism map onto the original ancient story of human creation?
And that brings us to the core synthesis of the text. The author directs our attention straight back to the book of Genesis, specifically Genesis chapter 2, verses 18 through 23.
The famous passage.
The narrative is universally known, right?
Oh.
God observes that it is not good for Adam to be alone. He puts Adam into a deep sleep. He extracts one of his ribs. He closes up the flesh in its place. And from that extracted rib, he forms Eve.
Right.
For centuries, this has been read as a pure miracle, requiring no mechanical explanation whatsoever, just poof. But the author asks a radically different scientifically grounded question.
Which is?
Is this surgical extraction biologically plausible based on modern osteology and genetics?
And the text's answer is a definitive, scientifically backed yes.
A yes.
To support this theological premise, the author introduces a specific, peer-reviewed piece of modern biological research. The text cites a comprehensive study directed by researcher Francesca Mariani at USC Stem Cell, which was actually published in the Journal of Bone and Mineral Research.
And this research, it really completely reframes how we think about the human skeleton.
It totally does.
When you think about losing a bone, you think it's a permanent structural deficit, right? If you amputate a finger, it does not grow back. The human body does not regenerate entire limbs like certain amphibians do. But the Mariani study reveals that the human rib plays by entirely different biological rules.
The findings detailed in the text are truly astonishing. The researchers observed that humans possess an extraordinary, highly anomalous capacity to repair and regenerate large portions of their ribs.
Wow.
Yeah. Unlike a femur or a phalange, a damaged or extracted rib can largely reconstruct itself.
But how? What is the specific biological mechanism the study identifies that makes the rib so unique? Why does it regenerate when a finger bone doesn't?
The text pinpoints a highly specialized layer of tissue called the perichondrium.
Perichondrium.
Yes. In anatomy, the perichondrium is a dense membrane of vascular connective tissue that acts as a wrapper around the cartilage of the rib.
Like a sheath.
Like a sheath, exactly. And according to the research cited in the source, this specific perichondrium is densely packed with highly potent progenitor cells, adult stem cells. These specific mother cells have an aggressive capacity to generate chondrocytes, which build cartilage, and osteoblasts, which build bone.
And the text highlights a detail regarding these perichondrium stem cells that is absolutely vital to the author's overall thesis. It states that these cells retain their regenerative capacity, quote, mesmo cuando dislocadas, even when displaced.
Even when displaced.
Wait, does that mean if you physically strip this tissue away from the ribcage and place it elsewhere, it will still try to build bone and cartilage? It doesn't require the microenvironment of the ribcage to function?
Precisely. That detail is the biological linchpin of the author's theory. The stem cells within the rib's perichondrium are so robust that physical displacement does not destroy their generative power. They are viable for extraction.
That is wild.
Furthermore, the text reminds us of another critical biological factor regarding osteology. The rib is a massive, highly active vault of cancellous bone.
Which means?
It means it is incredibly rich in red bone marrow. And as we established when we broke down the scientific triad earlier, bone marrow is the body's primary, most abundant reservoir for multipotent hematopoietic and mesenchymal stem cells.
Okay, let's clearly connect the dots for the listener here because the synthesis the author is achieving is just brilliant. The argument is that the creator, within the parameters of this ancient narrative, did not simply reach down and arbitrarily select a random bone.
Right. He didn't extract a metacarpal or a vertebra.
Exactly. The author is arguing that the creator utilized the singular bone in the entire human skeletal system that is perfectly engineered for a massive cellular extraction. A bone wrapped in a highly unique tissue densely packed with progenitor cells capable of regenerating the structural damage.
A bone that is literally hollowed out to serve as the body's largest natural vault of multipotent mother cells.
The text is basically asking us to view the Genesis account not merely as a theological metaphor, but as the first recorded surgical extraction of biological material, a massive, perfectly targeted stem cell harvest.
It's mind-blowing when you think about it. And when you analyze the ancient text through this very specific modern scientific lens, it profoundly recontextualizes the famous biblical dialogue that immediately follows the procedure.
Right, because right after the surgical extraction is complete and Eve is formed and brought to Adam, the text focuses on Adam's immediate reaction in Genesis 2:23. He says, "This, at last, is bone of my bones and flesh of my flesh."
Which is such a famous line.
For thousands of years, theologians and poets have interpreted this as the first romantic prose, right? It is seen as a beautiful poetic acknowledgement of their partnership and relational equality.
But what's fascinating here is how the author's biological framework completely transforms our understanding of that sentence. According to the ultimate conclusion of the text, Adam's exclamation is not merely ancient poetry. The author argues it is a literal biological statement of fact.
An empirical observation.
Exactly.
He is essentially waking up, performing a biological audit of the situation, and confirming the genetic reality aloud. He is recognizing that Eve's raw physical material, her foundational cellular structure, was quite literally extracted from his own biology.
The text emphasizes that, within this integrated framework, Eve is a direct, literal biological extension of Adam's body.
She is genetically distinct as a female, yet her foundational material was clonally derived from his extracted stem cells.
Exactly. And this creates a profound theological and genetic synthesis. The author points out an unbroken chain of physical creation established in the text.
An unbroken chain. The text maps it out sequentially, right? Humanity's raw physical form is initially assembled from the elements of the earth to form Adam.
Right.
And then the sequence continues directly from Adam's own biology to form Eve.
And the author argues that this specific chain, Adam from the earth, and Eve subsequently derived from Adam's potent stem cells, reveals a profound genetic unity inherent to the human species.
A fundamental cellular oneness.
Yes, a oneness that modern genomic science is only just beginning to trace backwards through mitochondrial DNA and chromosomal inheritance. The author concludes the deep dive with a really powerful moment of reflection, noting that this ancient narrative, delivered to Moses over 4 millennia ago, utilized accessible, seemingly simple language to embed a reality of surprising scientific precision.
The text actually concludes with a direct statement of awe based on this synthesis. It says, "Blessed be the Creator, whose wisdom precedes our greatest discoveries by millennia."
It's a beautiful way to wrap up the argument.
It is such a compelling way to engage with a text that is 4,000 years old, to look past the surface-level simplicity of a rib and to see the staggering blueprints of modern cellular biology, stem cell extraction, and epigenetic manipulation hiding in plain sight within the ancient prose.
It forces a total reevaluation of how we approach the intersection of science and scripture. At least from the perspective offered by the author, scientific discovery does not inherently invalidate or compete with the ancient theological narrative.
They aren't enemies.
Far from it. Instead, in this specific case, modern biology potentially provides the exact, tangible, mechanical structure for how the theological narrative was physically executed. The electron microscope and the ancient scroll are ultimately describing the exact same event.
To bring this all together for you, the listener, let's just trace the arc of what we've explored today. We started by looking at the monumental achievement of mapping the human genome, unlocking the 3 billion base pairs that act as the script of life.
Right, the alphabet.
We dove into the microscopic metropolis of the human body, understanding the complex epigenetic mechanisms that lock 216 cell types into their specialized roles.
And we analyzed the necessity of the biological backup system, the mother cells,
Mhm.
and detailed the intricate biochemical signaling that allows them to regenerate tissue through chemotaxis.
Right. We examine the scientific triad of stem cells, from the fleeting pluripotency of the early blastocyst to the targeted multipotency of adult bone marrow and the lab-induced time travel of iPS cells.
We also impartially navigated the profound ethical dilemmas surrounding embryonic research and the definition of human individuality.
And finally, we explored the text's breathtaking central theory that the Genesis narrative of Adam's rib perfectly describes a deliberate, highly accurate stem cell extraction. That the choice of the rib utilized the body's most regenerative perichondrium-wrapped vault of mother cells, making the creation of Eve a literal biological extension of Adam's own cellular matrix.
It really is a profound, seamless synthesis of empirical scientific reality and deep theological conviction.
It really is. And I want to leave you with a final thought to mull over as you go about your day. A thought that builds directly on the themes of this incredible source material.
Let's hear it.
Think back to the time machine concept we started with. Right now, modern medicine is pouring billions of dollars and unimaginable human effort into harnessing stem cells. Researchers in pristine laboratories are desperately trying to perfect the biochemical signaling required to coax a blank slate cell into becoming functional cardiac tissue, or a healthy liver, or repairing a severed spinal cord.
It's the absolute pinnacle of our medical science.
Exactly. We view this as the absolute bleeding edge of human achievement. We view it as stepping into a brand new, uncharted scientific frontier. But if the theory presented in our source text today is true, if the creator utilized these exact same biochemical mechanisms, these exact same pluripotent mother cells harvested from a human rib to orchestrate the formation of an entire complex human being, does that mean our most futuristic, cutting-edge regenerative medicine isn't actually a new frontier at all?
Oh, wow.
Are we not pioneering a new path, but rather is humanity just slowly, meticulously, blindly reverse engineering the very first biological surgery ever performed?
That is just a staggering perspective to consider.
It really makes you think. Thank you so much for joining us on this deep dive into the code of life. Keep questioning, keep exploring, and we will see you next time.